Cell functions are tightly regulated. One example is the regulation of the cell cycle, which if allowed to continue uncontrolled can potentially lead to cancer. One common type of signaling pathway starts with the G-coupled receptors. This animation shows one example leading to the activation of a transcription factor. We begin with the G-protein coupled receptor, or GPCR, which has 7 transmembrane domains. An extracellular signaling molecule, or ligand, binds to the GPCR, activating it and causing a conformational change that sends a signal to the trimeric G protein. The trimeric G protein has 3 subunits: alpha, beta, and gamma. The alpha subunit has a nucleotide binding domain, which holds guanosine diphosphate, or GDP, when inactive. When the protein receives the signal from the activated GPCR, the alpha subunit trades its GDP for guanosine triphosphate, or GTP. This is called a nucleotide exchange. The activated alpha subunit then dissociates from the activated beta-gamma complex. These activated components can now regulate the activity of target proteins in the cell membrane, such as the membrane-bound enzyme adenylate cyclase. Once activated, adenylate cyclase catalyzes the formation of cyclic adenosine monophosphate, or CAMP, by removing 2 phosphates from ATP. CAMP then acts as a secondary messenger by migrating away from the membrane and activating protein kinase A, or pKa, in the cytosol. pKa is a kinase, which means that it transfers a phosphate from ATP and covalently links it to a protein. Prior to activation, pKa is a heterotetramer consisting of 2 catalytic subunits bound to 2 regulatory subunits with CAMP binding sites. CAMP binds to these sites, causing a conformational change that releases the catalytic subunits and activating pKa's kinase activity. The catalytic subunit can then enter the nucleus and phosphorylate target proteins such as the transcription factor CAMP response element binding protein, or CREB. Once phosphorylated, CREB binds to specific promoters in the genome and starts transcription for cell growth. To stop the signal for cell growth, the alpha subunit will eventually hydrolyze its GTP into GDP with the encouragement of a GTP-ACE activating protein called regulator of G protein, or RGP. The alpha subunit then releases adenylate cyclase, which stops catalyzing the formation of CAMP. The alpha subunit returns to its resting state, recombining with the beta-gamma complex. The bound signaling molecule will often diffuse away on its own over time. However, if the receptor is active for too long, it may be phosphorylated by a GPCR kinase. Once phosphorylated, the protein arrestin may bind, causing the protein to be endocytosed and potentially degraded in the lysosome. If the cell did not have a way to turn off this signaling, the cell would continue to be activated, which could lead to uncontrolled cell growth, a characteristic that could lead to cancer.